Hello, welcome everybody. So we are back to our Wednesday webinars. These webinars are conducted by Pune River Revival. We mainly work for rights of rivers. We want rivers to be clean, flowing, naturally beautiful with rich biodiversity and good riparian forest. And through these webinar series, we want to reach out to as many people as we can and let them know about the importance of rivers, the riparian zone, trees, and the rich biodiversity. So today we have Kedar Champhekar with us, and Kedar is an expert in biodiversity who has been helping us out with various mappings, and he has done a lot of bird counts and lot of studies on the Pune river banks also. So I think it would be interesting to know from him how he actually measures the biodiversity and what are the important aspects and how do you actually quantify that. So Kedar, over to you.
Great. Thanks, Prajakta. I hope I'm audible. Yeah. Welcome everybody to this evening, and thanks for inviting me to give this talk today. This is not my first time giving a talk here, and it's always a pleasure to talk to all of you because we have such a vibrant community of people who are so passionate about conservation of the river. So it is always great to talk to all of you. So I'll briefly introduce myself for those of you who don't know me. I have a master's degree in biodiversity, and I've also done my postgraduate diploma in natural resource management at the Ecological Society. And for the past 17 years, I've worked in uh, basically worked in biodiversity education, conservation, content development, and many other capacities. Currently, I freelance as an ecologist. What I basically do is biodiversity assessments, mainly for environment impact assessment projects, but also for restoration projects as well. And I'm also been helping recently with the faunal biodiversity mapping of the Mula River, and hopefully will be getting involved in more faunal biodiversity assessment on the rivers in Pune. And I'm also involved with a lot of education initiatives with the Ecological Society, with Empress Garden, and other organizations in Pune. So today, what I'm going to talk about is quantification or measurement of biodiversity. Um, I have tried to make it very simple. So, considering that most of us here are lay people, people who are not from the field, and also people like me, probably who are afraid of anything to do with quantification. I am always been, when I was in school at least, always afraid of maths. So anything to do with quantification would scare me. But so I've just tried to talk about why we should quantify biodiversity. So we go out, we just look at the biodiversity, we look at the species, we'll write down the names, probably we record that. But going beyond that, some kind of quantification and measurement is necessary. So we will talk about why it is necessary and how to do it. So in how, I will cover mostly what are the techniques to do it, what are the different ways in which we can measure biodiversity, and then what are some of the very basic measurements, in the sense very basic, what you can say, indices or measures of quantification of that biodiversity. Samir is here, so hopefully Samir should be able to contribute more. Samir deals with a lot more data than I do. I think Amit Raj is also here. If he is there, if him and Prashant can also chip in, because they deal with data. I don't really deal so much with data, but I do deal with surveys. So I will talk to you a little bit more about the surveys as well, the way of surveying, the how you can survey. So let me share my screen, and then we will take it forward. And we will have the questions at the end. You can put it in chat. So at the end, maybe Prajakta can take all of our questions, all the questions. Yeah. Can you see my screen now?
Yes.
Okay, great. So let us look at measurement and quantification of biodiversity. And so let us start with the very basic question of why. Why do we need to quantify? Very, very simple. Most people might ask you, why do we need to measure? I thought that we can mostly divide this into just two basic reasons of why we quantify. One is to compare, and the other is to evaluate or to find out the value of something. So, comparison, very simple. You want to compare the biodiversity between two different places or the biodiversity of the same place over time, the changes that are happening in biodiversity because of time, or even changes that are happening because of any other kind of factors. So, for example, if you take the example of Pune itself, again, because we all live in Pune, we live in an ecotone, and we live in a natural, ecological, or even a climatic gradient. So, if you go west from Pune, you go to, say, the Panchet catchment, or you go to Tamhini, you have very high, heavy rainfall, 4,000, 5,000 millimeters, and you go, which is just 40 kilometers west of Pune, and you go 40 kilometers east, say to Baramati or Indapur, you have very little rainfall, 400, 500 millimeters of rainfall. And this rainfall gradient is natural because of the Western Ghats, and this creates a gradient in biodiversity. Now, if I ask you, you will be able to tell me, what the differences are, or in the ecosystem, say in Tamhini and that in Baramati. But quantifying it makes it makes it easier to demonstrate that difference. So obviously, if you go to Tamhini, the number of species in the forest in Tamhini are going to be more. Generally, in places with high moisture, higher rainfall, the diversity is higher, and drier places, the diversity is generally comparatively lower. I'm not saying that it is bad or it is low, but there is a difference, and there is also a difference in the types of species. So in Pune, we have this gradient, and if you want to actually show this gradient or prove that there is this difference, we will have to measure, or we will have to quantify that biodiversity. So comparison is easy, but other places where we want comparisons, especially right now in the context of rivers of Pune, is what happens before and after a certain project. For example, like the riverfront. So what is there before, and what is there after the destruction that happens? So that before and after, also you need comparison, and for the comparison you need to quantify. And the other is to evaluate or to value something, or to find out the value of something. So we don't need to, so people like us who are like-minded, who are pro-conservation, for us, nobody needs to come and prove the value of something to us. We already understand the value. But many times that value needs to be proven, either to bureaucrats, to decision makers, to governments, to funders, to all these people. Sometimes you need to prove that value, that, this is worth so much. That valuation can be in different ways. For example, it could be in the terms of carbon sequestered. So recently, we presented the tree mapping data at the Citizen Science Conclave, where Prashant had actually quantified how much carbon we are sequestering through that, how much carbon that those trees are sequestering. Now, this valuation becomes very important today, these days, because governments pay a little more attention when you say that, okay, this much carbon is being sequestered. And also, now there is this, probably carbon credits will come to India. Whatever we might think about that system, whether it is good or bad, you need evaluation in terms of carbon sequestered. The other type of evaluation might be directly in terms of money. So something that is directly quantifiable in terms of money. It could be goods, simple goods that we get from a forest. Say, for example, how much forest produce do we get from forests every year in India? And that quantification is available. You go to the forest department website of any state forest department, they will tell you, okay, this year so many crores worth rupees of forest produce was collected and sold. So the market value. The other is also the value of services. We'll come to that, which is much more harder to do. So, comparison and evaluation are two, reasons why we need to quantify. But what are these steps in quantification or measurement of biodiversity? Citizens like us, for example, when we get together, our biggest hurdle is, where do we start? How do we now prove that this ecosystem is valuable? How do we convince someone that this ecosystem is valuable, and how do we gather that data that is required for that quantification? So the first and foremost is objective. So the objective of quantification is very, very important, and it is very important to have a very clear objective, because otherwise you will go ahead collecting data in a way or in a manner that may not be suitable for you. For example, right now we did measurement of literally every tree that is present on the river banks. Why? Because what is our objective? Because we know that tomorrow, when now, and we are now seeing it, that when the municipal corporation has published its data, saying that there are so many trees and these are their numbers and these are we are going to cut and these are we are going to transplant. We know that there are going to be discrepancies in the data, and because of that, we need our own data, and so we need to measure literally every tree that is there on the ground. So this is a very, very intensive method of sampling. It takes a lot of time and effort. I'm sure a lot of people here know that because they participated. So because here the objective is to literally map every single tree. Okay, but that may not always be the objective. If say you want to find out that in Rammula confluence, for example, what is the diversity of trees that is present there? For that, you may not, literally sample every tree that is there. You might take a sample. I'll come to that. I'll come to what a sample means. Also, in the objective, you also need to specify how much time and resource do you have. So if you have very little time, you need to sample very rapidly, and your technique will again change. If you have a lot of time and resource available, then you might do more intensive sampling. So it is important to know the objective. The other is, what are you sampling? What are you looking at? So the taxon or the type of life or the type of ecosystem that you are looking at. For example, at Mula we have started looking at two things primarily to start with, and we will go to more. But one is, of course, trees and other flora. The other is fauna, in which we have started with birds. So if I say birds, then I kind of have to, when I say that I have to measure birds or measure the diversity of birds, then my technique has to be fixed like that. We also have to measure fish. Now, there are completely different techniques for measuring the diversity of birds and the diversity of fish. So once I know, I fix the kind of taxon I'm looking at, then my method is going to change. Okay. So that is the other thing, that the taxon or type of life or ecosystem. The other is area. How much is the area that I'm looking at? We might just be looking at the riparian forest of Mula River, say from Wakad to Sangvi, or the entire Mula River. Sometimes you might be looking at a much larger area. So the area will again decide the effort, how much you are going to have to sample. To give you an example again, I might be studying the biodiversity or the, say, floral diversity of just Tamini Devrai. Okay, so that is it. But somebody might be studying the tree diversity of the entire northern Western Ghats. Now, the entire northern Western Ghats is a very, very large area. So for that, I will again, need much more time. And then I will have to decide how much time I have and then how much sampling I can actually do. Then, of course, technique. So technique depends on how much time you have, how much area you are covering, and what kind of taxon. So are you measuring plants? Are you measuring within plant? What are you measuring? Are you measuring herbs or shrubs or trees or what is it that you're measuring? And also your ecosystem type. So, you might be measuring grassland versus you might be measuring a forest. So again, your technique will differ. Or if it's a wetland, or if it's, say, you're looking at an aquatic ecosystem, then again, your techniques will measure. Then after you decide the technique, then you actually go for data collection using that technique. To give you an example again, for the bird counts, whoever was there with me, what we did was we did these counts of birds, 15-minute checklists at different points along the river. So, that is the technique we used. We collected the data in some kind of a format. So then you collect the data, do data entry, and then you do analysis. So analysis where we will look at indices. What are these indices and what analysis we have to do, and then interpretation. So what does that analysis mean? So these are all the different steps in quantification or measurement of diversity, and we have to follow these steps. We can't just directly go and start collecting data without having an objective, because then we might not collect the data in the right way, right? And then it might hamper our entire exercise. So that objective is very, very important. So then what is this sampling? We keep talking about sampling, data sampling. What is the sampling? That are we doing? So in most cases, for example, measurement of total biodiversity of an area is impossible. For example, if I look at birds of Muna River, it is impossible for me to count each and every individual bird that is visiting the river, or each and every species literally that ever visits that area. It's probabilistically it is impossible. I mean, it's I won't ever be able to capture that in a survey. So in most cases, it's a sample. So it's a smaller, either a representative area or a representative population that is selected. So we will look at what are these sampling techniques. So you choose a smaller area, and you choose a part of the population that you are trying to study. So that is called as a sample. Now, of course, the bigger the sample, the higher the number of samples, the data is going to be more accurate. That's obvious. But it may not always be possible to sample that much. So, again, it takes us back to the objective. What is our objective? If our objective is to map every single tree, then we literally are not sampling, we are covering the whole area. But if our objective is to just say look at the ecosystem structure or the associations of species, then we might just do a few quadrants. We might say just sample 10%, 5% of the area, and we can say that okay, this is a representative of that area. Now, of course, there are ways of how you sample, in the sense where do you take a sample, right? So, but this is called sampling. And sampling has to represent all habitats. So if I'm saying that I am sampling for floral diversity or along the Muna River, then I have to take into consideration every habitat that is there. So there might be different habitats. There might be forest, grassland. Majorly we see these two habitats, forest and grassland, along the banks of the Muna River.
Wetland?
So I have to, wetlands also. Yes, there are also wetlands. So I have to then consider all these 3 or 4 different kinds of habitats, and then I have to ensure that I'm sampling all of these habitats. And I might decide how much I'm sampling based on how big these habitats are. If forest is the biggest habitat, then I take more samples in the forest. If grassland is the biggest habitat, then I take more samples in the grassland, right? Ideally, 10% should be sampled, but it's almost always impossible to sample 10%, because even 10% is huge. So if you consider even the Ram-Muna confluence, there 10% might be possible, but even there it is quite big. But if you consider the entire Muna River bank from Akad to Sangwe, which is 9 kilometers plus 9 kilometers, 18 kilometers, say of length, width is say maybe 100 meters or 50 meters, even 10% of that is huge. So we can try to sample as much as we can based on the time and other resources that we have. Sampling sometimes can also be done along an ecological gradient. So sometimes the habitats may not be very sharp. So at Muna Mutha, for example, if you go to RMC, the habitat demarcations are quite sharp. So the forest ends abruptly, then there is, the grassland on the bank of the river, and then there are patches of wetlands. But sometimes it might not be like that. Like I told you about the rainfall gradient along the Western Ghats, sometimes the variation is gradual. And if it's gradual, then you might want to sample along that gradient. To give you an example of this, uh, if you have been to the seashore, if you've gone to the seashore and you've gone to a rocky seashore, say in Mumbai, for example, you see these barnacles on the shore, on the rocks. And, according to the tide level, there are different species of barnacles. So at the place where the exposure is the maximum, which is the most away from the sea, there the rock is exposed for a longer time. And the closest to the sea, the rock is exposed for a shorter time. And actually, different species of barnacles live along that. So you might want to then, if you're looking at diversity of barnacles, you want to sample that entire stretch from the highest tide line to the lowest tide line to get a proper sample of that. In wetlands, you might want to do that right from shallow water up to the driest area of the river bank. So that gives you the gradient of moisture along which the flora can change. For example, Vadun's trees or Panjambu, you only see them closer to the water. And there might be some other trees which you see them a little up from the water line because they might not tolerate flooding as much as Vaduns or Karuns or these trees do. So that is a gradient. So you want to be probably sample along a gradient. So what are these 10 techniques? So what are these different techniques that you use for sampling? So there are many different kinds of techniques. I've tried to have just a very small basic list of techniques that are used, ecological techniques that are used in field for sampling. So the most easy is transects. So if you know what a transect is, this is a transect. So you basically walk through an ecosystem. So this is an example that is used mostly for trees. So if this is a forest, you assume that the green square is a forest. You walk, say, 100 meters, and in a width of 5 meters, you sample every tree that you encounter. And so that is a transect sampling. You can do this for animals also, or for birds. For example, you walk along 100 meters, the width will be more. Say you record every bird that you see in 20 meters on each side. So then that becomes your a bird transect. These can also be used for mammals, that you actually drive along a road in a national park and you note down every mammal that you see on both the sides, and you also note down the distance at which the mammal is, approximate distance at which that mammal is from you. So this is a transect method. It's the most common method used. It's a very good method for rapid sampling. When you want to do very quick sampling, you don't have much time and you don't want to quickly, just understand the ecosystem. To give you an example, I volunteered for this study long ago when I was in my MSc, which was about looking at the tree diversity of northern Western Ghats. And the entire Western Ghats, you can imagine, was divided into these grids. These big grids, I think of 5 meter by 5 kilometer by 5 kilometer grids. And in each grid, we had to take a transect of 1,000 meters by 5 meters. So we literally had to go to each grid, walk in the forest for 1,000 meters on each side, 2.5 meters, 2.5 meter, measure every tree that we come across. So this was done for the entire Western Ghats. I'll share you the link of some of the publications that have come out from that, if you are interested. So this is transect sampling. So what we have done for Ramuna confluence or for the Muna river bank is we have done a transect, uh, basically, but our transect is as wide as the river bank itself. So we have not sampled a smaller area. We have literally treated that entire Muna river bank as a transect and measured every tree that is present in there. Then there is quadrat. The quadrat is this. Quadrat can sometimes be used for a little more intensive sampling. So you can see this person here. What is he measuring? He's measuring herbs. So he's measuring smaller flora. So sometimes you can do small quadrats for flora, especially for herbaceous flora. You can also do quadrats for trees. They are a little more intensive when it comes to sampling, but when you want to do intensive sampling, generally a quadrat method is used, where you randomly place a number of quadrats in the forest area that you want to sample, and you measure every tree that is present in that quadrat. So for trees, generally for a forest area, a quadrat of 10 by 10 meter in very dense forest or 20 meters by 20 meters quadrats are generally used for trees. And ideally, what you have to do is you have to keep sampling. You have to keep sampling quadrats until you have at least 3 quadrats in a row where you don't find a new species. That's called species accumulation. So when you stop finding new species, that's an indication that you have probably sampled enough that now you're not finding any new species. So quadrat techniques are a little more intensive. So this is a quadrant technique. Then what you have is a point count technique. Now point counts are generally not used for flora. What I also want to tell is quadrats can be used for other things also, not just flora, but also insects. You can sample insects in leaf litter within that quadrant. Then there are point counts. So point counts is what we did for the Mula River, where you stand at one place and you decide that a radius of visibility. So you say 20 meters, for example, or 25 meters. At the Mula River, we just said that whatever is visible. So maybe in like a 50 meter or 25, 30-meter radius, we sampled everything. And you decide the time. So 10 minutes, 20 minutes, whatever. And depending on the visibility, you decide the radius. In an evergreen forest, for example, your radius is lower. In a grassland, your radius can be higher. So that is called a point count. Point count is mostly the most common technique used for birds, especially for birds when they're stationary, say like a wetland. So in wetlands, especially point counts are, most useful. Then you can also do trapping or catching, which is not generally recommended. You need permissions now for trapping or catching, but there are certain animals which you can't otherwise sample. For example, moths. So you put a light trap, you put a big light, you put a big sheet of cloth at night in a forest, and you look at whatever is coming into that trap. Or what you can do is there are nets which you sweep through vegetation and you catch insects. So then you look at how many different kinds of insects you have caught, you record all of it, and then you let them go. Invasive techniques are not used so much anymore. They were quite common earlier. For example, vacuuming insects from vegetation. So they will obviously die when you vacuum them, but then you know how much diversity is there. So for fishes also trapping has to be used. So you have to go with a net, catch the fish, measure it, record its species, and then let it go. So you have to do trapping or catching. Then camera traps. Camera traps are now used for larger animals, especially carnivores. So for tigers, it's a very common technique. So tiger censuses that are done in India are used are done by using camera traps, where there is a camera that is fixed to say a tree or a pole, and then the camera is basically emitting a radiation like which is cutting across the pathway in front of the camera, or it has a sensor. So basically, it's like a motion sensor, like we have motion sensing lights. So anything that passes in front of the camera, it will snap a picture. And it does that for tigers. And tigers can be identified because, like we have unique 5 fingerprints, the stripe pattern of each tiger is unique. So the number of tigers that are captured in the photos, you can look at the stripe pattern and say that there are so many different types of tigers. Then there is bird ringing. Bird ringing is used for other things, where you put a ring on a bird and then you see where it used to be used mostly to see where that bird goes. So if it's recovered somewhere else, the ring has a code. So that person who's recovered that bird can then communicate back to you. These days, instead of ringing, radio callers are used or radio transmitters are used to find out, especially the migration pattern. And then DNA analysis is also used. So in tiger census, for example, DNA analysis is also used. So it's used by extracting DNA from tissue samples. In case of tigers, the scat is used, or the excreta, the poop of tigers is collected, and then in lab it is analyzed. And then what measurements are you doing? So along with just species. So for example, we know this, those who have done the floral sampling, we know that now we don't just record the species of the tree, but we also record the girth, height, etc. Yeah, in terms of animals, we can also measure the length. In terms of, say, reptiles or fish, you sometimes also have to measure the number of scales or the type or the size of scales on the body of that reptile. So all these measurements are also there. So girth and height we record for trees because we want to find out the biomass and then the amount of carbon that the tree is sequestering and also the age, for example. So this is measurement. This is classic measurement of a bird. This is a dead bird. No more these days. This was quite common in the older days of ornithology. Now nobody kills a bird, but you can still catch it and measure it. So sometimes people put up nets, very fine nets to catch birds, and when they catch, they take these measurements of the birds. So sometimes you need measurements for different reasons to find out what's the average size of a bird in terms of, in case of chicks, you want to find out how fast they are growing or things like that. So we come to quantification. So what are these different measures of quantification? So how do you now quantify this diversity, this data that you have? So the most simple measure is richness, species richness. The easiest way of expressing biodiversity in terms of a quantity is species richness, which basically just means the number of species in an area or landscape or an ecosystem. Say, I think for Mula River, we have some 89, 90 species of trees or something like that. I don't remember exactly. Or we have, say, more than 100 or 100 and some 100. How much is it, Sanjeev? Do you remember? 110 species or something? I am forgetting. How many? 112, I think, species of birds that we have recorded. So that's the species richness. Period. That's it. It's a number that there are 112 species of birds, say in Ramula confluence, or there are whatever 80 species of trees in Ramula confluence. Simple. The simplest thing. Depends on purpose of study. Many times it can be sufficient that you are just measuring richness. Okay, but the richness also depends on the sampling effort. So if you have not sampled enough, then you probably have not captured the what you can say the all the species that might be present there. So the more you sample, the better you will understand what is the richness, species richness in a certain area. Beyond richness, there are certain other very basic quantities that sometimes are very useful. So there are three very basic quantities: frequency, density, and abundance, which can tell you about the characteristics of a community, or which can help you know something about that community, or it can help you compare. So frequency is what is frequency? You can look at the formula. It is the number of sampling units or quadrats in which a given species occurs. So how much? So if you have taken 10 quadrats, how many out of those quadrats say I have found currents? So that is the frequency. So if I found in all, then percent frequency is 100, obviously, right? So that is frequency. Density is the number of individuals per unit area that are found. So I might have found currants trees, and I might have also found something else. Say for example, I don't know, umber trees. So I might have found currants trees and umber trees in all the quadrats that I've sampled in Ramura confluence. But there might be far more number of currants trees as compared to umber trees. So just frequency might not give me the full picture. So I might look at density, and then but the density of umber might be much less than that of currant. So I can say that okay, currants and umber are both widely occurring in the area, but there are far more number of currants trees than there are of umber. And then there is abundance. Abundance is total number of individuals in the quadrats that the individuals occur in. So frequency, density, and abundance gives you are three very basic measurements that can tell you something about a community, and you can even compare species within the community. So if you want to look at what is, say, the dominant species of trees at Ramura confluence, then frequency, density, abundance will give you a very good picture. So, for example, currents might be having the highest density at Ramguna confluence because it occurs everywhere and in many and in quite a number, uh, right? So these measures can tell you something about an ecosystem. You can even look at frequency, density, abundance of different species and compare. So say you want to compare Ram Mula confluence with say the Pauna and Mula confluence, then you might look at both of this and then you might look at which species has the highest frequency, which has the highest density, which has the highest abundance, and compare it across these two ecosystems. And this will also tell you, this can also, give you some means of comparison, between the two, between different ecosystems. So these are some very basic quantities which you can use. Species richness obviously is very simple, but frequency, density, abundance is generally measured to understand the community in an ecosystem. And then we come to indices. So biodiversity indices. So biodiversity indices are basically, it's an index, or these are indices that quantify biodiversity, not just depending on the number of species which are there, but the proportional abundance of the species, which means what, say for example, how abundant is each species compared to the other. So like I said just now, umber and currants might be present everywhere, but currants might be far more abundant, okay, compared to umber. And also it looks at how evenly distributed the species are. To give you an example, below I have just given two tables. In table A, there are 4 different species, and these are the numbers. So species A has 10 individuals, species B has 4, C has 6, D has 4. And below that, B, all four species are the equal number of individuals in that. So B is considered to be more diverse than A, even though they might have the same number of species. Now, an ecosystem like B is hypothetical completely. It doesn't exist. It never happens that all the species in an ecosystem, all of them have equal number of individuals. That doesn't happen. It's just an ideal state. But the more number of species and the more evenly they are distributed, the higher is the diversity in an ecosystem. So, for example, in a place where only one species shows an extremely high abundance, might not be a very healthy ecosystem, even though there might be a lot of species in there. To give you an example, you might have an ecosystem where, say, on the Mula River, you might have a patch of forest where you find many species of trees, but mostly you find subabud, which is very, very abundant, which has invaded into that area. So if that subabud is extremely abundant there, it kind of, kind of overwhelms all the other species, then that might not be a healthy ecosystem because there is only a single species which is highly, highly dominant. And that generally is not the case in nature. Usually there are a couple of species that are, almost equally dominant. To give you an example, again, if we say go to, for example, Tamini, you might find several species that you see together and which are almost equally abundant. Say, which is Anjani, which is Nemesilon and Zambul. And then there might be a third species, like Pisa or something like that, which are equally, sort of equally or similarly distributed. So there are two biodiversity indices that are used commonly. Now, I will not go into the details of the maths of this. Even I am not very conversant with that. But there are two indices. One is called the Shannon index. So this is the formula for the Shannon index. So H dash is equal to minus summation of pi into natural log of pi. Now, what is pi? Pi is basically the proportion of individuals belonging to the ith species, which means for every species, if I have 10 species, if I go back here, in say A, the total number of individuals are actually 24. So for A, that proportion Pi is 10 upon 24. Right? Like that, I'll have to do for everything. So for B, it is 4 upon 24. For C, it is 6 upon 24. For D, it is again 4 upon 24. And then for each one of that, I have to calculate this quantity. So Pi into the natural log of Pi. And then I have to add all of this together. And then that will give me the value of H dash. So basically what the index is doing is it's taking into consideration the proportions of individuals of or the proportions of each species. So it is taking into consideration the evenness or the abundances of the species, not just the number of species that are present. Now the value for Shannon index varies between 1.5 to 3.5, generally rarely exceeding 4.5. So generally it doesn't go beyond 4.5. 3.5 is 1.5 to 3.5 is usually the range. In healthy ecosystems, you will find it closer to 3.5, or sometimes in very, very healthy, undisturbed, untouched ecosystems, it can exceed, beyond 3.5, it can be 4.5 or even more. And there is another index called Simpson's index, where this is the formula where again n is the I think n is the what is n is the number of individuals of each species, and capital N is the total number of individuals. And generally, it is expressed as 1 minus d, because otherwise for d the value becomes 0 to 1, and 0 becomes higher diversity and 1 becomes lower diversity, and that confuses people. So it's expressed as 1 minus d. So then higher the value, the higher the diversity, and the value for Simpson's index is between 0 and 1.
Okay,
I think for
pi, the
yes,
first
yes.
thing that says pi, is that by mistake?
No, not as it's not pi, it's summation.
Okay.
It's the summation, which means that you have that n into n minus 1 has to be
No,
calculated for every species.
summation I understand. The first
Where is pi?
point below
No,
that.
that's not pi. Sorry. Sorry. I carried it over from the previous one.
Yeah. That's
That's
what I thought. So
pi.
I just wanted to
Yeah, so
confirm.
instead of pi, it has to be n. Yes, I'm sorry.
Right.
Yes, here it has to be n, it's not pi. So basically, n is just the number of individuals in each species. That's all. So just ignore that first line. So 0 to 1 is the value of Simpson's index. Now, if you want to go deeper into the maths of it, you can go. There are lots of resources available. I'll even send you links to some resources if you want to go into the mathematics of the whole Simpson and Shannon's index. But for everyone, what is to be remembered is why these indices are important, because they don't calculate just the number of species, but also the proportional abundance or the evenness between the species in an ecosystem, because that is also important and that also tells you the health of an ecosystem. So that is why Shannon and Simpson indices taken into consideration, or Shannon and Simpson indices are important. I think if Prashant is there, I think for the Ramuna study that we did for the trees, the Shannon index is I think around 2.5, which is good actually. So it is above half of the normal range of that. So that considering that ecosystem is not very undisturbed, 2.5 diversity index is actually not bad. It's actually quite good for the RMC plus Mula Bank, the tree mapping that we did. Now going beyond that.
Okay, it's 2.25, Prashant says.
Sorry?
It's 2.25 for RMC.
2.25. Yeah. So it's actually good. It's not bad, I would say. For the amount of disturbance that is there in those ecosystems, the diversity index is still quite good. And so this is just when we are quantifying the biodiversity. So this is all we are still looking at the amount, the number of different kinds of species are there and the individuals are belonging to each species. But what about ecosystem services? Ecosystem services come from this assemblage of species. So how do you have an index which takes into consideration the ecosystem services? Especially, if you're talking about cities, there is a very, very good measure which is called the Singapore index. It is also called the City Biodiversity Index. And the City Biodiversity Index covers a very broad range of indicators from species diversity to ecosystem services that are offered by the ecosystems present in cities. So it's a very good tool for cities to measure and monitor biodiversity and ecosystem health. Now, the maximum points, so this index is in terms of points, it's measured in terms of points. So the maximum points you can get is 112. And PCMC, mind you, actually did this exercise a few years ago and they scored 58. unfortunately, I was trying to access the page where that report was there. So earlier you could actually download that report from the PCMC website. Now it says page not found. But I think we can try to find that report. You can even do an RTI or something to see if we can actually get that report from PCMC where they've done the Singapore index for entire PCMC.
So is 58 good, bad,
No,
average?
it's not good now. Out of 112,
No, I
uh,
understand, but I'm just
yeah.
thinking that in terms of how Indian cities are in the.
Oh, that way, not many Indian cities have done this. I think the only other example I know is Mira Bhayandar, and I don't know what is their index. So we actually this exercise needs to be done India wide. The other thing in this index which the PCMC did was I think it also considered all the defense areas in PCMC, and that kind of helped it in certain indices. But the defense areas are defense areas; they're not really managed by the PCMC. So if you exclude that, then the index might come down because the PCMC has no role in managing all these defense green areas which are with the defense. So the
Right.
green areas that are present in the PCMC's jurisdiction have to also we have to see that. So PCMC scored around 58. Now these are the measures. There are 28 different indicators against which you have to measure, and there is detailed methodology. I'll actually share the report. The whole document about the Singapore index and how to measure the Singapore index is readily available. This had come out of the UN United Nations Conference on Biological Diversity. I think in 2009 or 2011, or I think around that time, this index had come out of that, and there is an entire document on how to measure this index. So you can see that it actually measures quite comprehensively not just the diversity of species, but even proportion of protected natural areas, proportion of invasive species. If you look at this, uh, you know, kind of purple portion, then see it covers ecosystem services. So regulation of water quantity and recreational services, health and well-being. Each of this indicator, there is a detailing of how to measure that indicator. And then it also looks at governance and management of biodiversity. So from 15 to 28, 13 indicators are actually based on how do you manage that diversity. So that is also really, really good. So 56 points, maximum points are given for that management of that biodiversity. So this is a very, very good index, and every city actually should do it. And if you can get hold of that PCMC report, I think that will be great. So I'll
Yeah,
also try on my part.
if we find that report, then we will know that the number is not really 58.
Yes, yes. And we can actually see on each of those indicators how is the PCMC doing.
Exactly. For example,
So
health
that
and
will be great.
well-being, those
Yes,
kind of things.
yes. Or for example, budget allocated to biodiversity, or policies, rules, and regulations, and things like that. And there
Right.
are quite detailed points given on how each of that index has to be measured. So it's very, very good. We should do that exercise. We should try and get hold of this report from the PCMC. If we know anybody in the environment cell or something like that, and if they can, if we can get a report from them, this will be amazing. So this is there, and but beyond this, there is also ecosystem evaluation, which is much more complex, where it involves quantification of ecosystems in terms of the monetary value of goods and services provided by these ecosystems. So in terms of literal money, in terms of dollars, how much does that ecosystem, worth? And this can be an effective tool, of course, in informing policy and making a case for conservation, and also for funding. A very, very good example of this, you can go, I'll also share the link for that, is the Aghanashini estuary conservation in Karnataka. So where this Aghanashini river, that originates in the Western Ghats, goes and meets the sea, Arabian Sea in Karnataka, in North Karnataka, and it forms an estuary with really good mangrove forests and a lot of ecosystem services that an estuary produces provides, including fishing for the local communities. And there was a, I think, proposal for a port or something like that in Aghanashini, where the locals fought, and also they were backed by institutes like the Indian Institute of Science and Dr. T.V. Ramchandran, who took the lead, and he actually did the evaluation in terms of the number of billions of dollars of the ecosystem services and the goods that the Aganashin estuary is providing. And that kind of cemented their case against the port. And actually, that whole the Karnataka government decided to actually uh, cancel the development in the Aganashin estuary. And Dr. TVR has also done evaluation of the for wetlands in Bangalore. So something that we are trying to see if we can do it for Pune as well. So ecosystem evaluation is very complex though, and it involves a lot of different measurements of a lot of different factors, including the monetary value in terms of, say, if it's a forest, then how much value is the wood, how much value is the resin, the fruits, the seeds which might giving oil, and all of these. So all these goods that a forest gives, plus say the number of say crores of liters of water, for example, that is percolating or that the forest is conserving, and all of that. So there are many of these things that you have to measure, and then you come at a value of an ecosystem in terms of dollars. So I was talking to I had the pleasure of talking to Dr. TV Ramchandran about Pune and about the Mula River, and about particularly these 9 kilometers of the Mula River, where the not nine, so actually the entire riverfront project of Mula and Mutha, so that 44 kilometers of development. And he said, a very rough estimate, but it is very interesting to know that he said that 5,000 crore rupees are being actually spent on that project. And 5,000 crore rupees is actually the value of the ecosystem there in that. So the actual loss is 10,000 crore rupees, because 5,000 crore rupees is what you are spending to destroy 5,000 crore rupees of ecosystem. So, the actual expenditure is 10,000 crore rupees. And I think some 1,000 or 2,000 crore rupees of additional per year services that are given by this ecosystem in terms of, all the ecosystem services, if they are measured of this entire ecosystem. So it is a very rough estimate in 5 minutes. He was just like, to give you an idea, but a much more detailed analysis is necessary to actually find out the value of that. So with that, I'll stop. I think I overshot my time a little bit. If there are questions, please feel free to ask. I'll stop sharing the
That is
screen.
okay, because it was so good, Kedar, that I didn't want to interrupt and tell you that we are out of time. Because
Yeah.
it was really a wonderful
Yes.
session, and a
Thank you.
much
Thank you.
needed
Thank you.
session. I had asked in the chat before, can we unmute Samir, please? I mean, can we make him
Yes. Yes.
co-host?
Yes.
I think I can unmute, but I couldn't make him a co-host.
Oh, okay.
Let me ask him to unmute, and then we'll take the questions also.
Samir has unmuted.
Yeah.
Yeah. Yeah. So Samir actually, has more experience dealing with data. What I've not touched here at all is, handling larger quantities of data and the statistical analysis that is required most of the times, especially if you're doing a research project. But I thought that there is too little time to go into the statistical, methods. Maybe we could have another session for those who are interested by Samir and Prashant to look at the actual statistical techniques and tools that are and tests that are used for quantification
Sure.
and measurement.
Samir?
Yeah, sure. No problem. No problem at all. This was a very nice presentation, Kedar, to summarize everything in such a short time. Yeah, very well put.
So there are many questions. Should we take them one by one?
Yes, please. And there are some comments by Samir in between. I think we should read out.
I will also look at that. Yes.
Great.
So one, of course, Saili has asked if in an ideal situation, should sampling and biodiversity study be done across different seasons? Absolutely. Ideally, it is absolutely necessary to do biodiversity studies across different seasons. In India, especially, we generally do measurements across at least 3 seasons. So monsoon and post-monsoon, which is like the winter time, so around December, January, and then a summer or a dry season sampling. But at least monsoon, post-monsoon has to be done, ideally. And quadrat square, which was in an earlier image, can be assembled and dismantled. Oh, well, Saili, you can make a quadrat out of anything you have. You can just use sticks to do the quadrat, you can use a measuring tape and just keep stones, or you can actually create a device like that person is using, which is probably made using some PVC pipes or something for measurement of quadrats. And Samir, do you want to talk about your comment about the environmental DNA? Samir?
Yeah. Yep. So sorry, I was muted. I don't know why I did. Environmental DNA, you can think of using latest tools and techniques to just you imagine taking a sample of water from the river and analyzing it and finding out what kind of animals or small plants also live there. So that is like a very layman term or definition to it. So you don't need to actually sample or look at the animals. You just take the water, you analyze it, and you find what diversity there is in the water.
How much water would you take?
Great.
People have done it using like 1 liter or something, or even less.
Oh, okay.
I think it is also the technique used for looking at microbial diversity, especially in soil and things like that, where you
Yep.
can't really culture these
Yep.
microbes.
Exactly. Yep.
Okay, great. There are some of more of your comments. Do you want to just elaborate on that?
Could you just read out loud so that everybody would know those?
So there is another comment where Samir said these measures can also be calculated for each sample or each transect. Yes. So are you talking about the frequency density or indices?
Again, he I think by default he gets
He's getting
muted.
muted by
Yeah, I
By
don't
default. Yeah.
default,
know.
yeah.
Yeah.
Also, yeah, so just to add to that is it you can basically calculate frequency for any sampling unit that you have, be it a transect, be it a sample, be it a point observation also. So it's
Yeah.
basically relative occurrence or relative
Yeah.
observations of what you have to the total observations.
Yes. Yes. Then Shannon value is usually used for comparisons rather than used by itself. Yes, absolutely. I mean, generally, standing alone, these indices mean less, but
Hmm.
um, uh, yeah, especially for comparisons, because you want to know, just you have a number, doesn't tell you anything. You want to compare it to, say, a healthy ecosystem or a degraded ecosystem or something like that, to then give you an idea of what is happening to your ecosystem. So comparison is, I think, one of the biggest, reasons why actually even these indices are calculated. Simpsons is easy, easy to calculate. Yes, you don't have to do the log in Simpsons index. PCMC is a liar. Well, it would be interesting to see how PCMC got some of these measures. There is actually a an agency called Terracon Ecotech in Mumbai. So they have actually done this exercise for the PMC. They were the consultants for PCMC on this.
It would be interesting to see the data because I know for sure they don't have arthropod data with them. So I don't know how they
I'm sure
calculate.
they don't have, but I think the index allows you to choose taxa. They don't say that you use every taxon, but you choose, I think, a certain number of flora of faunal taxa or something like that.
All right. Yeah, of course, obviously. Yeah.
Yeah. So I don't know. It might have done, say, butterflies or odonates, but
Yep.
not,
I
say,
think so.
probably all
Yeah.
entire arthropod diversity.
Yeah.
And there are certain things I saw like education and things like that. They must have given higher points to themselves.
Yeah. So, it also all depends on what data PCMC shared with the consultant.
Right, right.
So,
Hmm.
yeah, yeah. So, yes. So, ecosystem services, like Samir has commented, are divided into provisioning services and
So
regulating
Kedar,
services. Yes.
we cannot see you basically. You haven't switched on
I
your.
know I haven't turned my camera. I will do that. Uh,
So
yeah.
at
Can
least
you see me
people
now?
can see who has been telling
Yeah,
them
yeah.
about this
Sorry, my camera is at a
biodiversity.
weird angle. You can see a lot of my beard from that angle usually. So,
No problem.
but hi everyone, this is me. And what is the method to calculate the age of a tree? Well, it's a little difficult. Um, you need the growth rate of a tree. For that, there is a way to calculate the growth through the age of a tree from its girth and height. But you need the growth rate of a tree, which is difficult to measure unless you have a plantation which I have been monitoring for a very long time. For tropical trees, especially, it is harder. For temperate trees, you know that method of counting the rings. And these days, you don't have to cut a tree to count the rings. You can bore a hole into the trunk and take out a sample and count rings. Because in temperate regions, what happens is trees stop growing in the cold season, and then they only grow in the warmer season. So there is a very distinct layer of new growth every year, which forms rings. So it is not always that prominent for tropical trees. So the ring method is a little difficult. But if you know the growth rate of a species, it's easier, or by comparison. And I think even carbon dating is used, isn't it, Samir? I think even carbon dating can be used to measure the age of a tree or of wood, or looking at
I
different
have no
isotopes.
idea.
Yeah.
I have no
So
idea.
But
yeah, but growth
actually,
rate. So the common method is using growth rate, and growth rate we don't have
Kedar,
mostly.
for our tree mapping, like our volunteers, hundreds of volunteers have done the tree mapping,
Yeah,
and they have measured the girth, and they have written the
yeah.
height by guessing it. So that should give a fair amount of, I mean, fair,
The
right?
problem is across species there is a lot of variation.
No, but if there are like standard, some of the species like currants, umber, and boar,
Yeah,
can
but
it
if
give
we
like
know if we can find out growth rates for them, we will be able to calculate the age. Yes.
so we would know growth rates for currants or no?
I am not sure. Probably not.
Oh,
Because those studies have to be done for growth rate calculation. Long-term monitoring has to probably be done. So I'm not too sure. We have to find out if there are growth rate of current trees readily available. We should find it out actually.
okay. Okay, great.
Sumit is asking, what would you recommend in terms of further biodiversity mapping along Pune PCMC river stretches? Some guiding documents on riverfront development suggest that a biodiversity survey should be conducted before the project. Uh, Sumit, the government has to do the EIA, environment impact assessment, and a big part of environment impact assessment is ecology biodiversity assessment, and that they have to do in the EIA. And actually, for the riverfront for Mula Mutha, we saw the EIA, and the EIA is completely, you know, like not properly done. So, for that entire 44 kilometers, they have said that there are some 18 species of birds, which is unbelievable. I see 18 species of birds from my balcony every day. So the problem is, if the government was doing their job properly, we wouldn't have to do all of this. So it is mandatory. And on the basis of the EI, also we can go fight in the courts. To give you an example, the Balbharti Paudpata project was challenged on basis of a faulty EI and a faulty biodiversity assessment, and then they had to redo the biodiversity assessment. So why we are doing this entire exercise is that we know that that data which is going to come out of the EIA is not going to be authentic.
Okay,
So
okay.
we have to do that. Yeah,
There is one more question about
yeah.
is there any record available for the restoration of the riparian zone anywhere?
Which riparian zone?
What he is saying is any riparian zone. Anywhere, is there
Oh,
any
any riparian
document
zone.
where we can
I'm
see how
sure there are
riparian
case studies.
zone was
Yeah,
restored?
yeah. I'm sure we can find case studies. There have been case studies of lake restorations or river restorations in India. I'll also find and send you a few case studies. So there
Yeah.
are case studies, I'm sure.
Please share with us so that it would be
Yeah,
very
yeah. I'll share it on the group.
important for us to know that.
Yeah. And also ecosystem services, why the authorities in the system can't understand while making such disastrous projects. Well, there are different things in that. One is, of course, vested interests because of which these projects are driven. The other is just lack of awareness. There is no training. So for example, training of IS officers, these things are just not there. And through ecological society is now trying to do this. to also reach out to institutions like Yashada, for example, where they do trainings of government servants, where you introduce curriculum in their training which talks about ecosystems, ecosystem services, and all of that. It is basically because they are not aware at all. A lot of times they're not aware. So one
Okay,
is
we'll
that.
take these, whatever are these last two questions we'll take, and
Yeah.
we really do not even have time to sum up also. So one is about rating PMC, PCMC in
I
the
would
docket.
not rate any age data as very accurate because, like I said, we have very little information on the growth rates of trees, and especially PCMC, PEMC data, because I've been working in the EIA field for quite some time, and I know that the ages are always downplayed because the amount of compensation to be given for an older tree is higher, and also for heritage trees above 50 years, you need more permissions. So age data for PMC, PCMC in their dockets is for sure to be suspected.
You will see
100%.
many trees age is like 48, 49, 47. You would see many
And
trees.
trying to go below 50.
Yeah. So
Hmm.
it's you can totally see that it's done
It
purposely,
is. It is. It is.
intentionally.
Yes,
And
but age data is very age accuracy of age is very. I will say if I say a tree is 50 years old, I will always say plus or minus say 10 years because there are so many other factors which also influence the girth and height of a tree. So the accuracy is low generally.
okay, so if you're saying plus or minus 10 now, here onwards it will be 38, 39.
Yes. So instead of age, so the whole flaw is in the tree act only, which is considering age as a measure of how important a tree is. We should just consider girth. That's it, irrespective of age. If the girth is higher, the tree is more important. So we should just do it according to girth classes. Forget age.
Okay,
Especially if you're talking about sequestration, we should just look at girth.
great. So
Yeah.
I
And
think there are
yeah,
one or two comments, but that is fine. They are not questions, so we can actually
yeah.
wind up now.
Yes,
Thanks,
yes.
Kedar. As
Thank you
always,
so much,
it has
Prajakta.
been so wonderful, and we got to learn a lot. And I know there is so much more to learn. And you
Yeah,
also
yeah.
share the couple of documents that you,
I will share some links.
and then I'll
Yes.
share it in the group.
Yeah.
And we'll see you again sometime. And we have these webinars every Wednesday. So if you have missed any of previous webinars, you can see it on our YouTube channel. You can go to YouTube and search for Pune River Revival. And next Wednesday also do come back. There is going to be another interesting webinar. We didn't get a time to talk about donations. So I'll just say quickly, if you go to the website puneriverrevival.com, there is a donate button. So please do generously donate so that we can do more of these kind of studies and biodiversity studies, because so far we have been doing voluntarily. But if we want to do more, then it is always good to raise some funds. Thank you again, and good night.
Thank you. Good night, everybody.